Hello. This is part 2 of chapter 8 that we are continuing our study of DNA replication, binary fission, and mitosis. So we had already looked at the very beginning of the chapter that explained binary fission. We've gone over DNA replication. You're going to want to make sure you review all the enzymes involved in that process, from topoisomerase that untwists the DNA, to helicase that unzips the DNA, to DNA polymerase that brings the nucleotides to the parent strand. Make sure you review the DNA strand being anti-parallel, with one strand running in the opposite direction of the other, and that it is a semi-conservative process that creates the new strand, with half of it being from the parent strand and the other half being a brand new daughter strand. So this second half of chapter 8 begins with the portion on cancer. So Henrietta Lacks is a very, very important person in the understanding of cancer cells. So this is way back in 1951. She had been diagnosed with cervical cancer, and while she was at Johns Hopkins, they had taken samples from her cervix, and the cells were incredibly received, because when they were put in the nutrient broth, up until that very point, human beings had not been able to have cells replicate outside the human body. So the minute they had these cells, and they very quickly started replicating, they knew they were very, very special cells. So they were named HeLa cells for her name, Henrietta Lacks. Today in 2021, we still have HeLa cells growing in the labs. So they're used constantly, and they have allowed us to learn a whole lot about cells and their behavior. So they're used to investigate cancer, viral growth, other processes in medicine. And these are just a couple pictures of Henrietta Lacks that are pretty famous now. A book in 2010 was written about her life, and your next discussion board is going to revolve around Henrietta Lacks, and you're going to hear her story. It's pretty compelling and interesting, and I think you'll enjoy this one. So, cancer cells, they're very abnormal. They look abnormal. Look at these cells, look at the blood vessels growing right to them compared to the normal cells. They're bigger, they're different color, they have blood vessels all over them, very different. One of the distinct features we know about cancer cells is they lose specialization. So when we say they lose specialization, remember when we were talking about differentiation, when you were a one-celled organism and then you all of a sudden were able to have bones and muscles and heart and nervous tissue, you lost that you gained a specialization, but cancer cells lose that specialization. They are just general cells that reproduce and wreak havoc in the body. They are immortal. They are able to continue to divide endlessly, which we'll investigate and explain, and they can regenerate the end of their chromosomes. And again, we're going to investigate that as we continue through the chapter. The cell cycle that we investigated in the first part, we will continue and look at a little closer. Everything we've talked about in the human body and a living organism is under very close, tight control. And the cell cycle is no different. So a cell doesn't just casually move through the cell cycle. There's actually a series of chemical checkpoints that regulate the movement of the cell going from phase to phase. So why would we need these checkpoints? So they're listed here. We want to make sure all the DNA is replicated. We want to make sure it's not damaged and that the chromosomes line up on the equator properly to be able to separate properly. So with that in mind, we want to recognize the interaction of gene expression with yet another aspect of life, and that is our cell cycle. So when the cell is approaching the point where it is going to divide, it's all about gene expression controlling that. Remember, the product of gene expression is a protein. So proteins have to be made and controlled that allow the cell cycle to advance or to just stop. So the differentiated human cells, when you first create a new cell, it begins in G1. And remember, every cell starts out in G1 and then they're shuffled over to G sub O. But notice there are some cell types that never ever move into the S stage. Those are your nerve cells and muscle cells. And you probably in the back of your mind knew something was different about nerve cells because there's people that get injured, whether it's diving into a pool or in a car accident or military individuals. If there's an injury to their central nervous system, they lose the use of portions of their body, whether it's their legs or their arms and legs or certain organ control. It's because the nerves cannot move, those cells cannot move into S phase and replicate. So once you injure those cells, they cannot grow back. Muscle cells are the same way, particularly of interest are cardiac muscle cells. When you have a heart attack, we don't like that because a heart attack actually kills cardiac muscle cells, and you need all of the muscle cells that your body has created in your heart to pump the proper amount of blood at the proper speed. So these cells that aren't allowed to move into S phase are part of the gene expression control over their cell cycle. These built-in checkpoints allow for problems to be corrected. And if you think about the trillions of cells that are in your body and that all of them are undergoing cell division for the most part, it's pretty incredible how few mistakes are made. We all know that mistakes can be made and that people have problems, and those problems can arise from cell division. However, the majority of them are A-okay. So it says a lot for the system and how it's able to correct itself. Now, there is loss of control. When that does happen, it's either going to result in cell death or cancer. So let's look at these checkpoints for a second. There's three major checkpoints. So G1, that's the one that controls whether or not the cell is going to progress into S phase, or it causes the cell to just stay where it's at. Then that there are cells that could possibly be evaluated and have damaged DNA. And so apoptosis is going to be allowed to go forward and they'll destroy that cell. The second checkpoint is G2. So the GAP2 phase checks for proper DNA replication that occurred in S phase. And it either will correct that and repair the DNA or signal for apoptosis. The M checkpoint makes sure the proper spindle assembly has occurred. It checks to see if the sister chromatids are attached to the spindle properly and if the chromosome alignment in metaphase is correct. So several checkpoints there, and makes sense that they would want to ensure that those work out right. But indeed, what if the body loses control? So we know everything's fine, you're healthy and all when we don't lose control, but what happens when we do? So the cell itself will just continue to divide. And they don't need to. Remember, cells get so big, they know their surface area to volume ratio is important to be able to move material in and out of the cell. And when cells override these checkpoints, they're allowed to continue to replicate. When enough of these checkpoint mechanisms fail, the cell loses control over its cell cycle and forms what we know of as a neoplasm. Neoplasm is just the anatomical word for a tumor. So a neoplasm has these cells replicating on top of each other. They're forming this great big large abnormal mass. In everyday world, we call it a tumor. So in this picture from your book, they're showing you, here is an uncontrolled cell mass that's been created. But what they want you to pay attention to is there's this capsule. So the capsule surrounds this uncontrolled cell population, and it would be defined as a benign tumor. It's contained, it is wrapped up in this capsule, or it's malignant. Nobody wants a diagnosis of malignant. That means that it's spreadable. This tumor is spreadable. Well, what do we mean by spreadable? Well, we use the term metastasize. A malignant tumor is likely to metastasize. And that simply means that cells are going to break away from the original mass, as they're doing in this picture. There is no capsule to contain it. And so they break off, and they either jump in a lymph vessel or a blood vessel. They can go either way. And once they're on the subway, they can go anywhere. So jump from the lung, and then they're showing you it went down here into the bone. Who knows? No one knows. It's totally random. A doctor can't control it. When you get to metastasis here, which is stage 4 cancer, we just don't know where these cells are going to jump out of the vessels into new tissue. That's what makes cancer so difficult to fight. And here is just a nice short little clip that you can watch metastasis.
The term neoplasm means new growth. Neoplasms may be benign, a growth that is generally harmless, or malignant, a growth that invades other tissue. Any malignant tumor is also known as cancer. Normal cells grow and function for a specific purpose. In cancer cells, the growth pattern is haphazard, has no specific pattern, and invades other tissue. Metastasis is the spread of the neoplasm to distant sites. Just the way plants send their roots into the soil, the finger-like projections of cancer force their way along the lines of least resistance and spread to other sites. The lungs are a common site of a carcinoma. The cancer cells invade the lung tissue. The cancer cells move from the lungs into the lymph system. As more and more cancer cells enter the lymph system, the cancer cells may spill over and enter the bloodstream. The cancer cells of the epithelial tissue, also known as carcinoma, metastasize from the lung into the lymph, then the general circulation, and metastasize to the liver. Cancer cells of connective tissue, also known as sarcoma, shown here in the thyroid gland, move directly into the general circulation. The cancer cells then metastasize to another site, such as the bone. Cancer of the epithelial tissue metastasizes through the lymph tissue and blood. Cancer of the connective tissue metastasizes through the blood.
Okay, so the doctors do know if a cancer arises in one type of tissue where it may metastasize to, but not exactly where, whether it will be the bone or whether it'll be the lymph, or whether it'll be right if it came from the thyroid gland, right? So that's what makes metastasis very, very difficult to deal with. And metastasis is a noun, metastatic is an adjective, metastasize is an adverb. So the same word, same meaning, just different forms of that word. Now, when we're looking at cancer cells, the thing that you have to, what we've learned over the past decades is how different they are from normal cells. And it's one way a pathologist will identify cancer cells, right? So of course they look different, and they can use the size from the picture at the very beginning of this section showed you two great big large cells that were identified as cancer cells with all kind of blood vessels compared to the natural shell, the healthy cells, and they were smaller, didn't have the blood vessels all over them. So indeed, they look different. You may have heard some people identify, they look at a mole on their skin, and a way to identify whether, people worry about whether it's going to be cancerous. They look at the edges of it. Is it growing up? Is it lumpy or is it smooth? So you look at the shape and features of these cells, and that helps you to learn something about the cell. The second thing is immortal. It ignores the normal cell cycle clock. Most cells divide 50 times, but if they have enough food and space, cancer cells can divide uncontrollably and eternally. And then the second thing under immortal is the telomeres, because this is where the cellular clock resides, is in these telomeres. And we'll talk more about telomeres here in a minute. But underneath telomeres is telomerase, and that's the enzyme that's needed to add DNA to the chromosome tips. And children make telomerase, or especially in an embryo, when you are establishing your DNA, you don't make telomerase as an adult. So we'll explore that here in a minute, but we're looking at the unique characteristics that make cancer cells different from normal cells. Okay, the third difference is resistance to controls over the cell division, and that's cancer cells lack contact inhibition. And typically when a cell is a normal cell, goes through cell division, it increases in cytoplasm and grows, and when it touches the next cell to it, it contacts it and it knows to stop growing and dividing, and they just know this is their space. So they exhibit contact inhibition. Cancer cells lack that ability. And the other thing cancer cells lack is what's called anchorage dependency. And anchorage dependency is ensuring that cell division occurs on a basement membrane or a basal lamina. Cells like to have their feet on the ground, and just like we do, but only it's called a basement membrane or a basal lamina in the body. Cancer cells don't care about that. They will divide on top of other cells, just craziness. So they lack anchorage dependency as well. And then finally, the fourth thing that makes them very unique is the ability to carry out angiogenesis. Angio is blood vessel and genesis is the beginning. So the beginning of blood vessel making is something that normal cells can only do for a short period of time. Cancer cells can stimulate it at any time, and they can grow blood vessels anywhere, in any place, direction, whatever. And that's why those great big cancer cells, I kept pointing it out in that picture. Oh, look at the blood vessels. Oh, they have blood vessels sitting on those blood vessels. That's the ability to create those blood vessels that ensures they have enough food and access to oxygen to sustain them. So that's still important. So those four things are very unique characteristics. Then finally, we'll go back to these telomeres here. The ends of eukaryotic DNA strands consist of these sequences that are called telomeres. They're non-coding, they're not a gene, they're not instructions of any kind. They're just simply non-coding portions that are on the ends of the DNA strands. That short little sequence they have listed here for you, TTAGGG. That is on the end of our DNA sequences, repeated thousands of times. Thousands of times. Why? Because it shields the end of the chromosomes from damage by normal cellular processes. Remember when we were talking about transcription, and I said that your messenger RNA transcript has to have a poly A tail. And the reason it has a poly A tail is because the enzymes and all these cellular processes in the nucleus have enzymes that are constantly snipping at their tails. They're after them to break down nucleotides. The same thing happens to our DNA when all this movement is going on. So when you have TTAGGG attached to the end of our chromosomes, it can snip that off. There's thousands of copies, and it won't damage the DNA sequence of a gene. So that's why telomeres are there, and that's why they're important. And every single time a cell divides, the telomere shortens. So it's giving information back to the cell as well. It may have divided 36 times. So the cell has information on how short that telomere is and how many more times the cell may divide. When they get too short, the checkpoint gene products stop the cell cycle, and it either induces apoptosis or it causes it to become senescent. And senescent cells are stuck in G sub O. They're metabolically active, but they don't function properly. And it may be just very similar to an organism or some structure that you know of. It still works, but it doesn't work right. It's just not functioning properly. And the longer it goes, the more progressively abnormal the organism becomes. So that's what a senescent cell is similar to. So most cells can only divide a certain number of times before they senesce. And that's what we believe sets the lifespan of the organism. So if, we say 50 times, well, the lifespan of the organism is probably around 50. But for human beings now, we're either believed to have a life cycle of somewhere to 78, 80 years old now. And so our probably cell divisions can go on approximately that number of times before apoptosis would be induced. Now, that telomerase stuff, when you're thinking about an adult now, not a child, and you're thinking about cells that can divide indefinitely, well, that would be our stem cells. And stem cells, we only have a few stem cells as an adult because you're not growing and developing anymore. But you do have to repair and replace. So you'll have some stem cells. That's what's in your hip joints. If you ever heard of somebody getting a stem cell transplant, they want to put a needle into the top of your femur, because that's where the bulk of your stem cells are. It's in the inside in the marrow of the bone. Well, why are these stem cells immortal? Well, they need to last your entire life for sure, but they're able to carry out this process because they retain the ability to make telomerase. And telomerase is an enzyme that reverses the telomere shortening that occurs after DNA replication. So this little enzyme in this pile of cells that you have in your bones still can make telomerase. So those cells are constantly made inside your long bone because they have this ability to make this enzyme telomerase, and they don't shorten the telomere. And that's indeed what we have realized is what is setting our lifespan. So cancer cells retain the ability to make telomerase. That's another thing they do, is they still have that ability, and that's why they live forever. So once we realize all what the differences are between cancer cells and normal cells, we know that mutations can occur as well in the genes. So if you have a mutation, and we know a mutation is simply the change of at least one nucleotide in a sequence of nucleotides, it could be one, it could be multiple. These mutations alter the function of the cell. And when it's a cancer cell, it's altering the cell cycle control genes, right? Cancer is uncontrolled cell replication. So it has skirted the control, the cell cycle control mechanisms. So there's two ways that cancer skirts around these control genes. So you can have an overactive proto-oncogene or an underactive tumor suppressor gene. Okay, so there's two types of genetic mutations that can occur. And it's all based on which cells are involved. If it's a somatic mutation, it occurs in a somatic cell. Remember, somatic cells are all the cells in your body except for your eggs or sperm. That's your germline mutation. Any cells that produce sperm and egg, if they experience a mutation, then they're going to pass that on to their offspring in the sperm or the egg. Now, cancer results from mutations in genes that control cell division. The mutation in those two types of these two types of genes are what can lead to cancer. If that proto-oncogene is mutated in any way, or a tumor suppressor gene is mutated in any way, the proto-oncogenes, they're the ones, we all have them, they're natural genes that stimulate cell division. So when cells need to experience cell division, a proto-oncogene is stimulated and goes into action. The tumor suppressor genes do exactly what you think. They suppress the tumor by inhibiting cell division. So we all have tumor suppressor genes because we don't want any of our cells to get crazy and start replicating uncontrollably. We don't want that cell division. So onco, that prefix onco means cancer. So oncogenes is any gene that helps transform a normal cell into a tumor cell. And oncogenes are created from proto-oncogenes. So when you have a proto-oncogene and something goes wrong with it, well, that mutation can turn your proto-oncogene into an oncogene. And that's why you have an oncologist. Oncologist is a cancer doctor. Onco is cancer. And you don't want mutations turning your genes into oncogenes. An example of a proto-oncogene that we all have is one that I put here. It stimulates the cell to divide and differentiate, right? So it encodes the epidermal growth factor receptor. So it's called an EGF receptor cell. That's an example of a proto-oncogene. We all need the epidermal growth factor receptor. We all need that. So as long as nothing goes wrong with it, it's fine. And we keep our epidermal growth factor when we're young, and everything is fine. But if something goes wrong, it can change that into an oncogene and stimulate growth. The tumor suppressor genes are just the opposite. They inhibit mitosis. So if you have a tumor that's forming, it tends to be a result of the tumor suppressor gene not responding to what's gone wrong. The products of these two genes, the BRCA1 and the BRCA2, these are the breast cancer genes. BR for breast, CA for cancer. The BRCA is usually what they're referred to as. The BRCA genes are example of tumor suppressors, and they will regulate the expression of DNA repair enzymes. The BRCA genes are the ones that would turn off in the cancer tissue any cell that wants to go through cell division that doesn't need it yet. It would turn it off. But if something is mutated and they can't function properly, then the tumor, the cell is going to constantly go through division and create the tumor. Now, these are the two that became famous when Angelina Jolie decided to get a total mastectomy. Angelina Jolie is an actress in Hollywood. Her mother and grandmother both died of breast cancer. She just had adopted a child, and she wanted to be around for this child. So she went through genetic counseling, had all kind of tests run, and it came back a 50-50 deal. There's a possibility you could get cancer or not get breast cancer. She elected to not flip the coin. She went in and had a total mastectomy, had all of her breast cancer tissue removed so that no tumors could result. Now, that's an example of an extreme reaction, and one, of course, that has money, because a lot of us can't go through the genetic testing that she was able to do, but it may very well have saved her life, but it's something we'll never know. There's also viruses. The HPV virus cause cells to make proteins that interfere with their own tumor suppressors. So you don't want to catch HPV either. There is a vaccine for that. So just be aware that there's some common things out there that maybe you didn't realize were involved with the reduction of cancer. Now, we all know that people that have cancer can undergo this is all about, okay, the DNA, your cancer risk. There's ways that we can reduce DNA damage that will ultimately lead to reducing your cancer risk. And eat healthy, right? They're showing you eat healthy, reduce your saturated fats, maintain healthy body weight. Tobacco has unequivocally been linked to lung cancers. Using a tanning bed is absurd. UV radiation. Self-tests, medical exams for early detection. Make sure you go and get yourself checked. Early prevention is the best. You don't want to wait till it metastasizes. That's stage 4, and there's usually no coming back from that. And then finally, avoid exposure to viruses, right? That's another way you can reduce your risk. And if you do have cancer, or you know people that have had cancer, or you watch on TV, indeed, surgery can remove some tumors. But if the mutation is already spreading throughout the body, you can't go in and do surgery and remove it all. So that's when chemo kicks in and radiation. It can radiation goes right specifically to spots, particularly in the brain. And as long as you can identify these cells and locate them, well, drugs and radiation will help. Drugs kill everything. When you take chemo, it goes in intravenously and it's killing everything. That's why you lose your hair, and some people lose their voice, and you're tired and lethargic and feel awful because you're literally taking a drug that is killing all the cells, right? It's very indiscriminate, trying to kill the cancer cells while you'll take you right up to the point where it doesn't kill you, but we're trying to kill just the cancer. And that's a tough call. Something that's becoming much more popular in today's world because we know more about genes and gene therapy, immunotherapy. This is the most promising because you're using your own genes and your own cells to fight the cancer, and they're genetically modifying your cells with the proper genes to stop the cancerous growth. So it's very promising, but not as widely available as we'd like just yet. But in the next generation or so, it'll probably be very commonplace. And then finally finishes up this chapter with bringing back the idea of apoptosis. It is programmed cell death, but to develop into the adult organism, we have to have a balance between cell division and programmed cell death. It has to be a balance. And when we talk about apoptosis, it's very different from necrosis. We're not talking about necrosis. That's accidental cell death, right? It's sudden, it's traumatic, it's very disorderly. Whereas apoptosis is very precise, coordinated events that dismantle a cell from the inside out, right? That's what's going on here. So you've got the receptor cell, or a receptor on the cell membrane that receives the signal to die. Here's the cell. This is apoptosis. Then executioner proteins and enzyme begin to break down the cell from the inside. It makes these big blobs and pieces of it start breaking off. And you have other cell components all breaking into pieces. Your immune system comes along and engulfs and destroys the pieces that are flying off. But don't worry, because if there's any proteins or nucleotides or amino acids in there, the body will reuse them. And they just have the example here of 2 toes on an individual's foot. This would be interpreted as webbing, like the duck and the chicken. And apoptosis failed to separate these 2 toes. Now, typically, a parent, when the baby was born like this would be very noticeable, and they would have the doctor split these apart immediately, and the 2 toes would have grown just naturally. But for whatever reason, they didn't have that done. But it's a good example to see how apoptosis had failed here, but yet it worked here. So interesting analogy here. But to look at the bottom line, apoptosis has 2 main functions: to eliminate excess cells, to carve out structures such as the toes, and to weed out aging or defective cells. And we need a balance of that with cell division. So that wraps up chapter 8. And hopefully you'll be able to get through this material and take your notes and refer back to it. And hopefully it'll be helpful. So thanks for joining me.